there will be many more IMBIOSIS webinar series, and I hope all of you will join us again in the future. So a brief introduction here. IMBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure that we are still connected with each other and stay motivated in our research despite the sudden pandemic. So as I mentioned earlier, today we commemorate the first joint IMBIOSIS APBioNet webinar. And today we are honored that we have a president of APBioNet, Associate Professor Dr. Muhammad Asif Khan, to be with us. And also we are greatly humble and honored with our speaker today, Prof Shoba Ranganathan, with I will read her brief summary later on. Without further ado, I would like to call upon Associate Professor Dr. Asif to give a few words before we start our webinar today. Hand over to you, Prof. Okay.
Thank you, Prof. Zeti, for the kind introduction and for the opportunity to be able to say a few words today at the Joint IMBIOSIS and APBioNETTALKS seminar. Can I be given permission to share very quickly? Can I do that?
Sure,
oh,
yes.
Okay, perfect. So I'm just gonna share a slide here. Can you see? Can everybody see?
Yes.
Okay, so I want to just briefly introduce APBioNETTALKS. So this is actually something that we just recently launched, and we are very proud of this initiative. And we want to, quickly. I just want to let you know quickly about what does it involve. So this is like we have many slides, so I just picked one slide that kind of sums up what this is all about. So. The unique value propositions include, it's meant to be inclusive, so it's open to anyone and everyone out there from the community who wants to contribute a talk, and even students, masters, PhD students who have something to share, they can come to us and tell us about what they want to do. And we will be able to make the arrangement for them to share to the community. It's open access, it's going to be free of charge. And it's meant to empower the community by sharing of knowledge. Of course, there's a lot already available on YouTube, but we felt that there's more that can be done, especially on tutorials, hands-ons, and even lectures, talks. There's just so much that's in bioinformatics that needs to be shared. And we have very limited out there, and more needs to be done. So we want to do our part in contributing to that pool of knowledge out there. So everything that we do, we will be recording it, so people who couldn't join can then see it later at their own leisure through our YouTube channel. So it's global. It's not, although we are APBioNet, which is focused on Asia Pacific, but it's open. It's meant to invite everyone from around the world to contribute, something of value to the community. So, so I'm really happy that we have already had 2 sessions, 2 talks done. This will be, we actually have one tomorrow, and today's talk is actually our first as a joint with a partner, and I'm really happy that the partner is IMBIOSIS. I am actually based in Malaysia, although I'm currently seconded to Turkey. So I'm really happy that this is starting from Malaysia and none other than IMBIOSIS. And what better way to make this happen than inviting our former president and our current chairperson of APBioNet Limited, Prof Shoba Ranganathan. So it's really a great pleasure to have Prof Shoba inaugurate this partnership seminar between APBioNet and IMBIOSIS. Thank you very much. And I look forward to the talk from Prof Shoba, and there's a lot that we can learn from her. Thank you, Zeti, and yeah, looking forward to the talk from Prof Shoba.
Okay, thank you, Dr. Asif, for your switch and short speech. I will give here some housekeeping rules for our webinar today. The lecture will take about 45 minutes, and the Q&A session will take approximately 15 minutes minimum, depending on how many numbers of questions that will be posed here. Okay, please submit the questions in the Zoom chat box and FB live page, and also we will have all these recording on the APBioNet YouTube channel. So during the questions session, I will read all the questions. Okay, and then before the seminar ends, I would like to invite all participants to switch on the camera for a photography session. And then please stay tuned until the end of the session to fill in the attendance form for e-certificate and eSpell credit. Okay, so today I would like, I am very honored to have our Honorable Professor Shoba Ranganathan from Macquarie University, Sydney, to be the first speaker for the first joint webinar between IMBIOSIS and APBioNet. So please allow me to share with you Prof Shoba's CV. So Prof Shoba Ranganathan is a professor and chair in bioinformatics at Macquarie University. And she is one of the women pioneer bioinformatician. And she also the first vice president for APBioNet. She has held research and academic positions in various countries, such as India, United States, Singapore, and Australia, as well as she has been actively involved for a consultancy in industry. So Prof Shoba's research addresses several key areas of bioinformatics to understand biological systems using computational approaches. And her group has achieved both experience and expertise in different aspects of computational biology, ranging from metabolites and small molecules to biochemical networks, pathway analysis, and computational systems biology. She also has authored several books as well as contributed several articles to Springer Encyclopedia of Systems Biology. She has a very wide experience in the field of bioinformatics and computational biology, and she has a lot of network. Okay, so without further ado, I would like to call upon Prof Shoba to share with us her interesting talk today on the benchmarking de novo genome assembly methods. Please, Prof, the floor is yours. Thank you.
Thank you. So can I share my screen? I'm very happy to be the first INBIOSIS speaker today for the webinar series into 2021. And I'm also honored that this has been co-shared as an APBioNet talk. I thank Professor Zeti and Professor Asif for their introductions. And I look forward to sharing some of our work with you. This is the work of a PhD student, and I will be happy to present this as quickly as I can share my screen. Okay. Oh, wrong one. Oh, just give me a second, please. I don't seem to have the right file up. Okay, what happened to my file? Right. I was working on a few talks and I think I got confused. So here we go. I can share my talk now. So share screen. Wrong one. Oh no, it says the wrong date and all that. So should be 12 March 2021. Okay, right. So we are looking at assembling a genome, and we would like to give you a very brief introduction, and then the lots of methods that we are looking at are reference genomes, the results, and the key points. I hope I have the correct PowerPoint slide. So genome assembly has been represented as a puzzle because we have so many elements that we have to put together. And in fact, in 2003, the British government actually published a stamp to commemorate the genome, which is called the end of the beginning, putting the puzzle of the human genome together. Genomics itself was a term that was coined way back in 1986 by the Jackson Lab geneticist Dr. Tom Roderick. And this is actually almost a decade after the large-scale genome sequencing methodology developed by Sanger. And this is now called the first generation sequencing approach, where fluorescently labeled tags were used to terminate primer sequences. And these colors were then observed using instruments. And then we get the 4 familiar genome sequencing label colors that we observe due to fluorescence, and that is what we see as our sequence today. A few years later, we have the Illumina or NextGen Sequencing coming up. A couple of methods are there, PacBio and Illumina. Illumina paired end sequencing is shown here, where we prepare a library of fragments with 3' and 5' ligated adapters. So we have the adapter, the insert sequence, and one more adapter here. And these sequences are added to the Illumina flow cell. Fragments are then amplified through bridge amplification in both forward and reverse directions, and clusters of reads are formed. Again, we incorporate fluorescently labeled nucleotides base by base, and then read the fluorescence signal for each cluster to determine the bases that were added. These are called NGS methods for short, next-gen sequencing. And similar to Sanger sequencing, they are constrained in the overall read length that can be produced at a reliable level. Nowadays, we have what we call third-gen sequencing, TGS. And examples of these are Nanopore and PacBio Hi-Fi. Nanopore uses a totally different approach, and I have shown this here in the diagram. It uses a double-stranded DNA, which is then split into a single strand of unamplified DNA and passed through a protein nanopore across a membrane, which has a fixed voltage applied across it. DNA single strand enters the pore with one strand passing through, and the current in the membrane changes based on what DNA bases are actually physically entrapped within this pore. This change is then detected using semiconductor sensors, which is also called base calling. So this is an absolutely new approach, and in this way, very long pieces of DNA are able to be sequenced. Again, this is called the ONT, Oxford Nanopore technology. Sequencing itself has now gone on to many other higher levels. We also have high-C reads, which form clusters and can help us to assemble longer fragments or even bridge gaps in our sequencing when we have consensus reads that don't actually completely overlap. And these are the most commonly used methodologies that we see today: Illumina and long reads with PacBio and Nanopore, and some high-C to finally bridge the gaps. And this has been reviewed by Gianni et al. In 2020. So please look for this review and you will be able to find it. So how then did we get caught up in all this? So we actually have part of a big grant which uses sterile insect technologies to get rid of pests in Australia. These pests are the fruit flies, Australian fruit flies, which were to be genomically sequenced. Phil Taylor, my colleague, is the lead author of this grant. I am one of the investigators representing bioinformatics, and Rahul Rane and others here from the CSIRO team are our partners who actually have a lot of expertise in growing these flies, extracting the DNA and sequencing them. And then we sit down and figure out what is the genome. This is quite different from Drosophila melanogaster, the vinegar fruit fly, which was sequenced way back in 2000. So this is the reason why we are involved in this grant. It was a very large grant from Horticulture Innovation Australia. And this actually is going to help us to control the spread of these fruit flies, especially because some of them, which were very tropical, are now moving into subtropical areas due to climate change. And this is a very big problem for Australia, which has a very large horticultural industry. So this was our basic plan. We would take all the available methods, benchmark them, develop a pipeline, and then apply it to all our sequencing that we do on the Bactrocera species, then do some comparative genomics, visualization and storage. Sounded very simple. These are very novel organisms that we are going to work on, very far removed from Drosophila. They are eukaryotes and insects, not animals. But each study that we looked at reported in the literature uses data that has different coverage and a whole different variety of tools. So it got to a point when we had to start with genome assembly, looking at the many different genomes that were sequenced for prokaryotes, but these were only done with long reads, no short reads or hybrid approaches had been used. A number of different methods have been used, and this was benchmarked in 2020 by Wick and Holt. But for eukaryotic genome assembly, there was no shortcut because there doesn't seem to be any benchmarking available. We have lots and lots of short read data. Generally, both short reads and long reads are generated. Many methods have been reported. Draft assembly also needs error correction, especially for long read data. And this is called polishing. And this can be done with data from the same or different sequencing technologies. And we also have a plethora of polishing methods. So finally, we assembled a list of how many methods have been reported in the literature. Because if you do a benchmarking study, we have to cover the field. So we came up with 20 assemblers addressing Illumina, Nanopore, PacBio, and hybrid data, as well as polishing. 8 different polishers can be used, and depending on the data you have, some of these names you will find on different technologies as well. But putting them together, we get 8 polishers, and these can also be used on different types of data and in multiple combinations. So we had the fruit fly sequenced, and then we now are going to benchmark and apply it to these Bactrocera. And this is what we're going to do. And we have applied it to 1 or 2 species already, but everything is, under comparative genomics, under review. And so finally, I guess we will publish sometime this year. So the data we collected for benchmarking are based on 2 model organisms, C. elegans, for which there is a lot of data, and also Drosophila melanogaster, which is an insect and therefore very relevant to us. And you can see that for these 2 organisms, we do have Illumina, Nanopore, and PacBio data for both, and the coverage is very variable. Very different for the 2 organisms. And we wanted to also see if there were any coverage effects. So we were going to randomly pick data from each of these at different coverages, and then see if there is also a minimum coverage that we require to get sequenced for eukaryotic genomes. These have effects on how much funding is required and how much data you need to produce, which types of data for the future for all genomic projects. How do we know that we have a good assembly? So we looked at the metrics for evaluating the results. And then we compiled what we thought were reasonably good methods. So we have assembly stats, which gives you the N50 value and the total assembly size. BUSCO is very frequently used because we look for gene completion as genes are reported by this score. BusComp is actually developed by a colleague who is in University of New South Wales, Rich Edwards, and BusComp completeness and identical scores are also calculated. There is also cost, which gives us details of SNPs and indels based on 100 kilobases. And then red gives you total repeat length, which is also very significant for eukaryotic genomes. And when we did the benchmarking, we specifically chose a single computing node with 20 cores running Ubuntu and with only 128 gigabytes of RAM. This actually tells us that this kind of small system can be found in many labs, and therefore we wanted to benchmark on that. We didn't go for a high-end multi-computer system or one of these proprietary systems, because then we cannot actually provide something that is useful for the entire community. In order to see what is the coverage effect, we subsampled the data using a script called BBMap. So this randomly generated 20x coverage, 40x coverage, etc. And then genome size estimates were for 100 megabase pairs for C. elegans and 140 for D. melanogaster as the baseline using the subscripting data software. So we generated data at 20x, 40x, and used the complete data set, full coverage for the two model organisms. And there were over 14,000 assemblies made with all the different software that we ran. And the input data was three types: Illumina, Nanopore, and PacBio, both for C. elegans and for Drosophila. We pre-processed them, then assembled them. We assembled individual data types and also ran hybrid assemblies. Then we did short read polishing, long read polishing, and then short read polishing, final with long reads and hybrid polishing, etc. And then we looked at all the metrics that we already showed you in the earlier table. Okay, so how did we do? So here we have the results for C. elegans assemblies, and we see here that we have the raw data and then the final statistics for each of the four types of runs that we carried out: independent runs for Illumina, Nanopore, PacBio, and the hybrid, and then for the final statistics after all the work was done. Illumina alone does not get polished because it is already high quality data, and therefore we have just transported these values in the second set of graphs for comparison purposes, and I put gray boxes to indicate that. All the rest were polished with 4 rounds of long reads, followed by 3 rounds of short reads. And short read here being the Illumina data. Overall assembly size tends to increase with coverage. So when we go from 20x, which is in red, to 40x, which is in green, to full coverage, which is in blue, we do see some improvement in the assembly statistics. And 2 of the programs could not assemble more than 60 to 70% of the genome at 20x, and these are Shasta and Falcon. And if you are using these programs, you do need coverage more than 20x. Long read polishers tend to overestimate the genome size, and you can see that they just start going upwards. We could see that the polishers were here, but when we did the final assembly and cleanup, that sort of corrected itself. Hybrid and Illumina are closer to the reference genome sizes. And the reference genome size was estimated again using those programs for comparison and also compared with the real results that we already have. So continuing on, we see that GC content and total repeat length are shown here. Once again, Illumina data was not polished. And for the other three, we noticed that 20x may give some errors or overestimation of both GC content and total repeat in the raw stage. But in the final stage, the total repeat length does come down to the baseline, although it's a little bit higher as far as the GC content go, but PacBio and hybrid data are very much in the ballpark. So repeats are not improved by polishing, and therefore that's something to be aware of. So we do know that Illumina and hybrid approaches are really doing well, and they are probably giving the best result. PacBio is not far off, but PacBio data, of course, is a bit expensive. I believe 10x is coming back, so that's something we haven't considered. So gene completeness for the C. elegans assembly was estimated by Busco scores, and they are quite good for Illumina and hybrid assembly. That's shown in graph A, but overtaken by long read assembly after short read polishing, shown in B, and also some data is here in E comparing the Busco scores initial to final. BusComp, the other metric I was talking about, is able to find more complete genes in the raw assemblies for long reads compared to Busco. So we are wondering if this is a better predictor of the final assembly quality right away with the initial raw data. So as you can see, BusComp is here, the scores, and Busco scores are here. So that's what we are looking at. This is also shown here. So the initial BusComp scores seem to be comparing well with the final assembly as estimated by Busco. And it looks like it's a quick way to figure out what kind of final quality you might arrive at. When we look at 20x, 40x, and full coverage, we see here that the Busco scores are definitely leaning towards longer coverage for Illumina. And Nanopore, some methods are doing better than the others. And the different methods have been shown at the bottom. PacBio is quite good, but the hybrid seems to be doing well throughout. So maybe if you choose the right programs, you can survive with only 20x coverage. It is a possibility. So when you go to hybrid, you do get good results. So low coverage can be polished up. Of course, it depends very much on the organism you have. But if you only have funding for 20x, that might be enough if you are able to select your programs carefully. So we see here that the best methods that we can see here are spades and soap. And fly for PacBio does very well. We also see that the top assemblies from cumulative z-scores, we have N50 that is getting higher and higher, higher the better. And all these data and tables are also available on GitHub. We have looked for complete Busco score genes here, and the higher the value, it will be better. Similarly, when we look at the percentage identical Buscomp genes, higher the value, that will be better. For SNP errors, we need lower numbers, and for indel errors, we need lower numbers. So the z-score looks at all these separate values as well, and then we evaluate it. So when we look at the cumulative z-scores for the top 3 assemblies for each data type, given that we had such a big matrix of methods to try out, we see here that Illumina is shown in peach, Nanopore in green, PacBio in cyan, and hybrid in purple. We see here from the cumulative z-scores that we actually find much better performance for the full fly, doing very well in all approaches, and Illumina is slightly low on the z-score side. If we look at the coverage measurements, 20x and 40x are here compared to the full. So that's what we could actually figure out. And the best method still remain Fly, especially Fly does very well on PacBio data for all coverage types. And Fly does very well for Nanopore, full, and 20x, slightly lower for 40x. So we could actually compare the different methods and the coverage data to figure out the best possible outcomes for each of the runs. Some of the details are here. For Illumina, Spades and Mazurka performed the best. Spades is here. And for Nanopore, Fly and Raven did very well. And for PacBio, Fly and Kanu did the best, but Kanu is extremely slow. So unless you have lots of time on your hands and can spare a big computer, it's perhaps not a good idea. And hybrid overall, Mazurka and Fly did extremely well. So the data is here, and it's a lot of data, but everything has to be sorted, and it's just a snapshot of the data has been shown here. So, and Hassler and PacBio do very well at 40x coverage, and DBG2 OLC does well at the lower coverage ends. So that's what we could get at. And for 20x, the best methods are shown here, and the number of times we need to polish, nobody has actually benchmarked that. We just did it till we got no more better improvement, and then we stopped. So we've got a different set of numbers when we come to the polishing strategy. When we look at melanogaster, applying the same methods, we're only showing you brief results. We again see that the Busco score is quite good for Illumina and hybrid raw assemblies. Here, that's what we see, the Busco scores. But overtaken by nanopore assemblies after short read polishing, that's shown here and here. Buscomp is again able to find more complete genes in the raw assemblies, that's here, Buscomp, and in the raw assemblies for long reads compared to Busco, that's shown here in D. Again, the Buscomp genes give us a very good idea of the percentage completeness of the raw assembly, similar to Busco genes in the final assembly D, that's shown here. And it could be a very good predictor of what we are going to get. The color scheme, if you've forgotten, peach is Illumina, green is Nanopore, PacBio is in cyan, and the purplish one is the hybrid in these diagrams. 20x coverage is in red, 40x in green, and the full coverage is in a shade of blue. And the different shapes indicate 20x, 40x, and full coverage. So we can see that we are able to get somewhere. And the top 3 assemblies for each data type were again compared. And again, we note that Fly is doing very well for Nanopore and PacBio data. Kanu does do very well for PacBio data, but only for 40x and full coverage, doesn't want to run or assemble at lower coverages. So we can see here that all the z-scores are positive compared to the previous one where the z-scores for Illumina were negative. But then I think we have more or different kinds of coverage for melanogaster compared to C. elegans, and that could be the difference. So to summarize, we have hybrid and long read assemblies generating more contiguous assemblies than short reads. This might improve with high C scaffolding, but not everybody does high C. So although we generated high C for some of our special Bactrocera species, we have not done any benchmarking there. Hybrid long assemblies look good, but long read assemblies after polishing are actually better. So maybe that is a strategy that we need. And the assemblers, how did they perform? For Illumina, Spades was top of the list, followed by Mazurka and RAP. W2 RAP was actually very fast. Nanopore Fly did very well. Raven was good after polishing. Shasta was at high coverages. Kanu is similar, but very, very slow. And similarly for PacBio, Fly and Kanu did very well. Also, Falcon did very well at high coverages, although it was quite slow. For hybrids, Mazurka and Mazurka plus Fly did well. Also Hasler, but only under high coverage and with PacBio data. Overall, the best was Oxford Nanopore and PacBio CLR assemblies, plus short read data for polishing that produced the consistently better hybrid assemblies, but it was a bit slow. Illumina only assemblies are also more accurate, but they are definitely more fragmented. So because they are only short reads, but they had a much better per base coverage. So Illumina is something that everybody should consider, and maybe one of the other long read methods, and then you would get a good result. So we feel that peak performers when weighing only key metrics were Spades and Mazurka for Illumina, Fly for Nanopore and PacBio, and DBG2LC and Hasler for the hybrid assemblies. Polishing is essential for long read assemblies, and hybrid polishing with hypo worked very well for ONT and PacBio. Long read polishing followed by short read polishing is also good. And then short read only polishing is sufficient for the best raw PacBio assemblies, such as those produced by Fly or Kanu. So depending on what data you have, you can actually figure out the strategy. We are also putting together a pipeline which, given the data, will automatically choose the methods and do the runs, and also give you the ability to run this. This is being set up because so many methods have to be put together and tutorials have to be made, but it's underway and it will be available maybe sometime later. Coverage can improve the assembly quality, but if you have only limited funds or are working on some strange organisms and want to do a pilot study, 20x coverage may be enough, but of long reads of either type with 20x coverage of short reads. And you would still get comparable quality with lot of savings in both time and money. And though this may change for more difficult to assemble genomes of totally novel organisms. So we are developing a pipeline called Pyro. It's on the way, not ready yet, and we have tried to apply it to Arabidopsis thaliana, and we've got some very good results from Mazurka, Fly, Again, fly for PacBio, fly for Nanopore, and Mazurka plus fly for hybrid, using the results from the benchmarking, and it's all getting written up. So that's all I have to say, because I wasn't sure how long this introduction would be, and also to give you some time to ask questions. So we do have some extra slides, but I think I will stop sharing and ask you for some questions.
Thank you very much, Prof Shoba, for the very interesting talk. So shall I start with a question?
Of course.
Okay, so I wonder, for the cumulative z-score, what are the component, or what are the parameters have been used to calculate this score, and what is the threshold to show that the results is acceptable? Thank you.
So the z-score,
Belagaknya. Oh,
please. Okay. All right. Okay, Prof. I can't hear you. What's wrong? Okay.
okay. So I think we need to share again, so I can show you the z-score. The exact calculation it actually uses a weighted thing. So in the paper we'll have the full details, but we used N50, percentage of Busco genes, percentage of Buscom genes, SNP errors and indel errors. And that's how we calculated the z-score. And I refer to the GitHub site. It might have the correct or the updated z-score calculation. But then with all this data, we could actually put together the z-score value. I'm happy to also post any details or further by email or email it to you. Thesis is under review, so I don't know if I can give you all the details right now.
Zeti, could I ask a follow-up
sorry.
question?
All right. Yes. Okay, please do.
Thank you, Prof Shoba, for the very insightful talk. And we ourselves are currently working on de novo assembly. So this was very timely. Thank you for that. With regards to the z-score, initially for Illumina, you showed that they were below 0. And then towards the end, they were positive. So sorry, maybe I missed something. What was done so that they were recovered? And why were they negative initially? Because you said they were...
No. They did not recover. Actually, the z-score is for different organisms.
Uh-huh.
So the first set of slides were for C. elegans, and that is still negative.
So is that concerning, the fact that z-scores were negative for Illumina when you showed that some of the best results were from Illumina? So kind of conflicted.
But the best results were from Illumina, but the best results were also very short. The assembly is very fragmented for Illumina. That is the problem.
I see.
Okay.
So I can show the slides again. Is that something you would like?
Yep, that'll be helpful.
Let me go and get my slides again. So share PowerPoint. Okay. So the results, I need
in
to
the
put
graph. Yep.
coming up. So the results here, this one is for C. elegans, and it does show negative z-scores for Illumina, but positive for all the others. And the second lot is actually for melanogaster. And here all the scores are positive. Although they are very small. But still, Illumina does very well. Only problem is the Illumina is very similar to this picture that I showed you from the review article.
Hmm.
The Illumina assembly is extremely fragmented.
Hmm.
It's very high quality, but only comes in short segments. Whereas the nanopore maps are much longer, and we are able to get much better contigs with both the short read and the long read hybrids. And you might have to do high C to actually fix the gaps in the long reads and the hybrid scaffolding. So that is the final step, but that's not been considered for the benchmarking. I mentioned this before, but if you have specific genomes, you can also do the high C scaffolding. So the results are different for the different data sets that I'm showing. There are two data sets, and sorry, I should just quickly go on to the results for you. This one was actually negative from the 40x and the okay.
Oh,
Shall I stop sharing? Is that okay, Asif? You're happy?
Thanks. Yeah, thanks, Prof. And maybe if,
There are
if
many
one more
questions
question.
in the chat box.
Sure. Just one quick one. What is the prospect of Nanopore being a dominant, sequencing platform moving forward, given that, it's longer reads and the benchmarking that you did? Do you think this could be something for people to consider, Nanopore?
Yes, at least you need one long read and one short read to get excellent results. That is our opinion. And Nanopore is definitely cheaper than PacBio unless you have a deal with PacBio. So I think maybe we could at least consider one long read and one short read to produce very good quality results.
Thank
So
you, Prof
that
Shoba.
is the way to actually go. Of course, if you can if you can do both, that will be great, and even 20x coverage at both using both technologies will practically give you a better quality output.
Thank you.
So
Thank you
one
so much.
lady has asked, what do I do for 20x coverage? The results are here. The assembly for short reads. The ones we found that did very well are actually Spades and Mazurka for Illumina data, and that would be the best method to use. It's also here. Spades and Mazurka. Spades was the best for Illumina for 20x, so that might be something that lady can consider. I saw that on the chat box. So that's something to use. Also, there is a whole list of methods, so I'm happy to answer questions separately if you wish. Is the assembly haploid? Which pipeline would be used facing a diploid genome? These are all diploid genomes, aren't they? C. elegans and this thing. The assembly actually comes out only as a single string, and we believe that the other DNA would actually be the complement. So that's how most of these methods assemble the genomes. Why are the results different between the organisms? That's because of the data coverage, and whatever data we got in the public domain, we have used. So that is the best we could find. We saw many organisms, but many have only two types of data, not three. And in some cases, the papers say the data is there and it's downloadable, but we could not find the data in any of the public repositories. It's the same with some of the recent papers. Everybody says they submitted data to the database, but there's nothing in the database or it's hidden. So we had to take what we got, and we chose C. elegans because it's got a lot of experimental information. Every gene is practically well established, and similarly, Drosophila has been well studied. Drosophila is also an insect and therefore relevant to our studies. But these methods should technically be applicable to most eukaryotic genomes. We are actually waiting for other pest organisms to be sequenced, and we are going to apply these approaches to see how they cope. That's why we applied it in the pipeline study to Arabidopsis thaliana, and we got very, very good results. I showed you some of the graphs. I didn't show you everything because it's another long process. But since we got that far, we think that we have a very good chance of getting the other unknown genomes also. Can we use genomics technology in plant crop improvements? I'm not actually a plant biologist, but I do have colleagues here who are doing genomics to improve plants, and I can put you in touch with the colleague Brian Atwell, who's here, and he might be able to advise you on that.
Prof, I wonder, one of the steps in your pipeline is on the data polishing, right?
Yes.
So would you share with us what are the steps that you use in polishing the data?
Yes, sure.
Yes, thank you.
We said so somewhere. I'm just trying to find it for you. Okay. All the polishing was done. No. I can't find the, just give me a second to find the thing. So. I think all of them were polished 4 times with the long read and 3 times with the short reads. And that's till they got consistency. So that's what we have done here. I'm just trying to find the here it is. Illumina alone was not polished. You can see that. Because Illumina is short reads and we don't polish it. It's never been polished. So we can polish it, but it doesn't seem to improve much. So Illumina is shown here. Rest were polished with 4 rounds of long reads followed by 3 rounds of short reads. So the same data can also be used to polish this, the data, and that's how the polishing was done. Also, different methods shown here take different types of data. Yeah. So not everything would take the same kind of this thing. So we had to actually choose methods and the data and pipe them through that.
Oh, please.
So I hope that is satisfactory. So we used 4 rounds of long reads and 3 rounds of short reads for the polishing, especially for the hybrid.
Okay.
So this is for the hybrid, and the rest we can actually polish using the same data again and again, and or try with the short read. So we have done both. And in the end, whatever we got, everything was then benchmarked, and based on the z-score, it was put together. That's how it is a cumulative z-score. So we just have to see which one has higher N50, so you score it that way. The bigger the number, it gets a better weightage, and then you score with higher BUSCO genes and then BUSCOM genes, and then lower, this will be negative SNP errors and indel errors. So that's how you get your cumulative z-score.
Okay, so Prof, with the system spec that you showed earlier, how long does it take to complete the whole process for the Australian fruit fly analysis?
Fruit fly is not shown here. So we have only shown here C. elegans and
Yep.
melanogaster for CPU.
Yep.
The fruit fly is much more complicated organism, and it's also a novel organism. Some methods didn't work. Some of the things had to be tweaked. But for C. elegans and nanopore, we have the CPU time here. And the methods here, some of them are very fast for Illumina, Nanopore, PacBio, and hybrid. Some of the methods are extremely slow, like DBG2OLC hybrid actually took about close to more than 7 hours for hybrid assembly, and Falcon PacBio takes a long time as well, 3 hours, more 3.5 hours for the full coverage. But many of the others are reasonable. In 4 hours, you can actually finish the work. Melanogaster took actually longer than the C. elegans.
Yep.
And we do not know why all these things take more time or less time. And for some of them, we did not show the data when it took too long. We just removed it from the graph. But Spades takes over 12 hours for hybrid. Falcon takes 12 hours for the PacBio data. Whereas for Nanopore and Illumina, you can easily manage under 4 hours for Nanopore and just over 4 hours with Mazurka for the raw data for Illumina. And this was for melanogaster. As you can see, this is 2 to 8, and this is already up to 16 hours. So each data set seems to require better handling. The coverage was not that different, but we have no idea why this takes so long or so little time. The methods also have been summarized based on this kind of study, based on the CPU time as well as the results. So we've looked at both. We also saw that memory usage can vary from program to program. The worst was Falcon, which PacBio provides. It actually requires lot and lot of memory. So does Raven for some of the data. And some of the older methods like Platinus, which is not very commonly used, but we benchmarked everybody. So you can see that they also require a whole lot of memory usage. Some of the other methods are very good. Mazurka does very well with limited coverage and gives good result. And Mazurka Fly is here and Spades is at the very end. Some of the programs did not run on some of the assemblies, which is why we have a gap here for the 40x. It is not only the options in running the code, but also different flags are required. And I think Pendy Tong had asked the question. I think all those details are actually provided on the GitHub website. Even how to run each of these programs to actually get an output.
So, Prof, this the results will be published soon, because I think the one that you did is
yes, we are waiting for thesis reports to come in before we.
Okay. Oh, that's good. All right.
So they might ask for more changes, so we thought might as well do everything when we submit the manuscript. So it's getting ready, and we are also doing some more methods, I think, for the paper.
Oh, that'd be awesome. Thank you, Prof.
So there are some questions I didn't answer. I'm trying to look at them. Work has not been published. So due to why to do hybrid between optical mapping and Illumina for short read. any two methods would actually give good results, and even 20x seems to give good results. So that's what we benchmarked here. So we could see that 20x was sufficient to get good results for these two organisms. And the pipeline has been set up and can actually run for any of these approaches. Yes, there are plenty of options. I address that. There are many flags also that need to be properly set for each of these programs, and that itself is a bit of a mess. But we can actually do that, and we have given on the GitHub site the commands for running each of these programs efficiently. We do not know the circular consensus sequencing method. We have not analyzed that data, but PacBio by itself seems to be a bit more on the high side of the budget. So if you can do the PacBio, that will be good, but you might need some support, and maybe you can benchmark the circular consensus sequencing versus the conventional CLR. So I hope this will encourage people to actually do more work and supplement what we have done.
Okay. So, Prof, have you answered the one due to money constraint? Is it to do wise to do sequencing hybrid? Have you answered that one?
Yes, yes.
Okay.
Yes.
All right.
I said the 20x, but using 2. That was in the last summary slide.
Yep.
So having 20x coverage of long reads of either type with 20x coverage of short reads may be sufficient to still produce comparable quality, considerably saving time and money. But if we are not sure, because we do not know what kind of organisms people are working with. If it's a totally strange or new organism, it might require more coverage, and I don't know how to predict that. I don't think anybody will be able to help predict that.
There is another question, Prof. Could you please explain why did the long read polisher tend to overestimate the genome size?
No, we really don't know why these things happen. And the polisher perhaps tries to assemble too many things, and it's overeager and tries to extend. So we do not know, because we are not developing in any of these methods. And if you read the papers for each of the methods, it's only been benchmarked against one data set. So this seems to be another problem that we face. We do not know, but that can actually be fixed with different rounds of polishing, or with using short read data that manages. We are able to actually bring that down to proper size. We do check with using BusComp and BusCo where we are compared to the reality, because for both these genomes, we have the full set of genes known. So that helps us to understand why we are actually getting overestimation of the size. It's a process of assembly. So I think that's the reason why this happens.
Okay. So what will be the challenges if you work on the human genome? Will be a different set of challenges that will occur? Prof, you unmute, please. Okay.
Okay, the human genome seems to have, we are not working on human genome because so many people have already worked on it. Much of the data that is available also, much of the benchmarking studies are for single chromosomes. We haven't done single chromosomes also because our main aim was to apply this directly to our pest genomes, which are insects.
Okay.
So that has been our aim in this study. But if somebody would like to do that, that would be a great thing to do. Maybe I was thinking human and mouse would be good, but it's a lot of data and a lot of coverage. and we are not really sure how much to tone it down. But it won't pick up things like variants and things, and people are already into multiple problems with the human genome. So maybe we should just focus on that. I don't know.
Exactly. Okay. Thank you very much. So I believe there is no more questions, but I think it is a very, very informative and useful knowledge that we have today, because I know there are many research in Malaysia doing on this genome analysis as well. Yeah, so we look forward to have your article published and use it as a reference. Okay, so I would like to thank you again, Prof. Shoba, for your time and your knowledge sharing with us today. And I hope to have you back again soon for your another interesting talk.
Thank you, Zeti. And I can confirm that the Z-score was simply, I think, an assembly of all the metrics that we talked about. It's just a simple sum with the plus and minus incorporated, because that's the way you can actually estimate it.
Yes, cool.
It wasn't a very complicated math, so there was no real formula or anything.
Okay, thank you, Prof. So, so I would like to ask all participants and speaker to on their camera for the photography session, so we can immortalize this session. So, right. So now we have actually during the talk we have about 2007 participants in Zoom and 28 participants on FB Live. So this is a very good turnout. Thank you very much, all participants. Yes?
207, not 2007.
No, 207, sorry. Okay. Okay, so maybe my team can do the Intan. Could you please?
Okay, ready?
Okay.
1, 2, 3, smile. Okay, another one. 1, 2, 3, smile.
Okay.
Okay, thank you.
Right. So I would like to thank all of the participants and also the technician in helping us setting up the smooth Zoom webinar today. Right. So then the last message from the housekeeping is like the participant can scan the QR code later. Okay, and then it is only available for 10 minutes. Okay. So then have a great Friday afternoon and a great weekend to all of you. Thank you very much and hope to see you all again. Thank you, Prof.
So can I leave now? No photograph?
Yeah, all done yet just now.
Okay.
Okay. Thank you very much.
Zeti, I hope I answered questions. I'm feeling rather sleepy now.
Yeah, I know. I'm sorry if I have to make you stay back at work.
Okay.
All right. So enjoy your dinner. Thank you.
Bye-bye.
Bye.
Everybody.
Thank you. Thank you, Prof. Shoba.
And thank you, Dr. Asif.